Hello, I'm Dr. Elizabeth Coon, and it's really an honor to be able to speak to the Defeat MSA Alliance Conference. Today, I'm going to be speaking on multiple system atrophy, touching on pathology and environmental factors. I have no disclosures. And these are objectives: to appreciate the naming and the history of MSA, to recognize the pathology and the multiple areas affected that's recognized in that name, and then discuss environmental factors. So first, let's talk about the naming. And so by employing this etymology and onomastics, the study of naming, we can get a better grasp of what we know of MSA, and by understanding that history often helps us with where we're going. The initial accounts of what is now known as MSA were in the late 19th century. French and German accounts talked about orthostatic hypotension with other movement disorders. But it was really in 1900 when Jules Desjardins and Andres Thomas described a syndrome called olivopontocerebellar atrophy with two patients, one of whom had an autopsy case. This was a 52-year-old woman and a 42-year-old man. And they had clinical symptoms of ataxia, dysarthria, akinesia, rigidity, hyperreflexia, and urinary incontinence, all of the clinical features we think of MSA. And on the pathology, there was atrophy of particular areas of the brain termed olivopontocerebellar atrophy, or OPCA, terms that are still used today pathologically. And then in the 1960s, a group of Van der Ecken, Adams, Van Bogart published a pathologic account of a patient with predominantly Parkinsonism and termed striatal nigral degeneration. Also in the 1960s is when Milton Shy and Glenn Drager published their 4 cases of autonomic failure with the classic motor features of Parkinsonism and ataxia. And it was right around this time when the Adams group published a case of a patient who had what they called both striatal nigral degeneration and olivopontocerebellar atrophy. So really brought the two pathologic features together in one patient. And so from that time on, there was a lot of the Shy and Drager syndrome that was used in literature. But in this 1960 article by Graham and Oppenheimer, they proposed using the term multiple system atrophy. And they say the similarities are obvious, and it would be unreasonable to attach different diagnostic labels to these cases, meaning like the striatal nigral and the olivopontocerebellar atrophy. And what they wish to avoid is the multiplication of names for disease entities, which are really just the expression of neuronal atrophy in a variety of overlapping combinations. So that's where we get the term multiple system atrophy. From that time on, there's a lot of different terms that were used in the literature: Shy-Drager, multiple system atrophy, and it was really with the consensus statement in 1998 where we see more of the terminology and the definition of MSA in the literature. In this consensus statement, they say these confusing terms, multi-system degeneration, should be discouraged and recommend more the MSAP and the MSAC rather than striatal nigral degeneration and olivopontocerebellar degeneration, which are more of the pathologic terms. And then we saw more of this consensus too with the 2008 criteria, which were used just until recently when we had the Movement Disorder Society criteria published in 2022. And so that's where we get to the naming of multiple system atrophy. And it touches a bit on the pathology, but I want to dig a little bit deeper in the pathological features of MSA. So let's start historically. So Frederick Levy published in a textbook Lewy bodies that were found in patients with Parkinson's disease, and this was in 1912. You can see these, fantastic pictures that he published. And then in 1997, there was really the breakthrough with the Spillantini and the Nature publications showing that alpha-synuclein was the composition in these Lewy bodies. And I just think it's striking that over this time period, these pictures are so similar from what Levy described to what we see under the microscope with alpha-synuclein staining. And so when we think about the synucleinopathies, then we know there are shared clinical features in this group with REM sleep behavior disorder, autonomic dysfunction or autonomic failure, and then the smell being affected more in the Lewy body disorders, but not in multiple system atrophy. And then an important part in defining these differences in the synucleinopathies occurred with Bannister. And again, you'll recognize Dr. Oppenheimer's name too. So they really looked at a lot of patients neuropathologically with synucleinopathies and helped break apart Lewy body disorders from multiple system atrophy. And this was publishing 16 cases, which delineated MSA from Lewy body based on pathological findings. Also, some notable clinical things that patients with Lewy body disease tend to have older age of onset. So pathophysiologically, we know that MSA is an alpha-synucleinopathy, but it is characterized more by oligodendroglial cytoplasmic inclusions in its difference from those Lewy bodies, which are what's seen in the Parkinson's disorders, Parkinson's disease and dementia with Lewy bodies. And we also know that there's differences in where this alpha-synuclein is deposited. So in MSA, we tend to see fairly central deposition of alpha-synuclein and these gliocytoplasmic inclusions. And really the brainstem in particular is very affected, especially that ventrolateral medulla, where we know a lot of our blood pressure control is from. There's also involvement of the spinal cord with relatively spared peripheral organs in multiple system atrophy. And I think this really gets back to the naming then. So we can see these multiple areas of the brain, the spinal cord, brainstem involved, which kind of gets with the naming, and then leads to the clinical symptoms that we see, be it the motor involvement or the autonomic failure that can present in our patients with MSA. And now I want to talk a little bit about pathophysiology and what we think is going on with the alpha-synuclein deposition causing damage in MSA. And I really like this figure from Dr. Svechuli and Venning, and they highlight possible genetic factors as well as environmental factors that are probably contributing to this pathophysiologic process that is causing the abnormal alpha-synuclein deposition, leading to abnormal misfolding of alpha-synuclein, and then that affects mitochondrial function, oxidative stress, and neuroinflammation. And this alpha-synuclein's story has really developed over the last several years, and in part because of some of the features that we now know of occurring in these alpha-synucleinopathies. And in particular, some of that is related to this prion-like spread of alpha-synuclein. So when we think of prions, and prions are proteins that are self-conforming, and then they become self-propagating. So they kind of are sticky, and they grab other proteins and cause these proteins to stick together. And there's probably specific strains of alpha-synuclein that are contributing to that prion-like spread. And so from this review from Jellinger talking about how this alpha-synuclein becomes misfolded, turns into an oligomer that affects a neuron, affects an oligodendrocyte that causes that gliocytoplasmic inclusion that I talked about. That then causes the oligodendrocyte to die off, and we have demyelination of the neuronal process, which leads to the hyperreflexia, the upper motor neuron syndrome. We also see that contributing to the degeneration of the neuron and these accompanying process of microglial activation and reactive astrocytosis. So I think that this story has really evolved in the last several years, and I'm excited for what that will do for further understanding this pathologic process in MSA, but also what we can do for different treatments or to stop the process before it really kills off these neurons and those oligodendrocytes. But another thing that I want to talk about next is how environmental factors may play into this pathophysiologic process. So what caused the alpha-synuclein to be affected in the process? And what is driving this microglial activation and these reactive astrocytes? And that's where the question of environmental factors come in. And specifically, is there an environmental trigger for multiple system atrophy? So there's been various studies reporting associations between MSA and different environmental risk factors, including organic solvents, plastics, pesticides, agricultural activities, metals, and smoking. I'm going to highlight a few of those and what we think that may be doing to contribute to the pathophysiologic process, the pathology in MSA. So a lot of these studies are older. So we're looking at a 1991 study that was published in Clinical Autonomic Research. It had 60 patients with MSA. And in this study, they showed that MSA patients had more exposures to metal dust, fumes, plastics, again, those things that I talked about as environmental factors. And then after that, too, in 1999, the Jankovic group out of Texas looked at 100 patients with MSA. It was all retrospectively looking back. And they found that of this 100 patients, 11 had heavy pesticide or toxin exposure. So I'm going to highlight those patients that they discussed then. So you can see that the clinical features all fit with multiple system atrophy. The occupations definitely varied, as well as the toxin exposures. But a lot of these toxin exposures that they reported in these 11 patients often happened over a number of years, including over decades, like a 30-year history, pesticides and malathion, and some of these pesticides that we know are problematic in other diseases. But you see, there's still quite a wide variety. And I also want to point out that in this group, too, they talk about other patients being affected, and some with, Parkinson's disease, others with, Shidrakar or multiple system atrophy. And that speaks to something that sometimes we hear about different clusters of MSA. And so when we're thinking about different environmental causes of MSA, clusters are important because if we see there's too many cases as would be predicted, just with natural variability, then that may give us a clue that something may be a potential trigger that has affected multiple individuals. However, it's very hard to do these cluster analyses, but something that is kind of important in these epidemiologic studies. So so that's kind of what had been done before, and bringing up that perhaps there are environmental factors that are triggering multiple system atrophy. So I'm going to share some cases that we saw. So this was a gentleman who developed imbalance at the age of 63, and then he developed progressive falls, bowels, and then bladder incontinence. He had dream enactment behavior, and on examination, he had Parkinsonism. It did not respond to levodopa. And his autonomic function testing, which is showed here, really showed severe autonomic failure with 63% anhidrosis shown in yellow on the thermoregulatory sweat test. He had a rapid progression of disease and died just 2 years, 3 months from the onset of his symptoms, with his autopsy showing the characteristic pathologic findings of MSA, namely those gliocytoplasmic inclusions. His wife was also affected. So her history is that she had dream enactment behavior and then began to develop urinary symptoms in the same year that her husband began to have his progressive decline. Her urinary symptoms progressed more slowly, but eventually she developed urinary retention and incontinence requiring a catheter. And then about a year later, she developed more speech changes. So her voice changed. She had a dysarthria. She was found to have central sleep apnea and then began having falls, and on examination also had Parkinsonism. Her autonomic function testing are shown here, again showing autonomic failure with orthostatic hypotension. She had a global anhidrosis on thermoregulatory sweat tests in a pattern that we frequently see in MSA, where there's preserved sweating over the hands and largely an essential pattern. She later, at the age of 67, developed severe stridor and passed away at age 68, with her autopsy also showing pathologic findings of multiple system atrophy. So that led us, to question, why is it that a married couple both had multiple system atrophy, if it is truly a sporadic disorder? And so we questioned their son, and the son said that um, they had maintained railroad tracks to a grain elevator. And in maintaining those railroad tracks in this Midwestern climate, used significant amounts of chemicals, including pesticides. And he listed all of the pesticides, chemicals, and elements that were in the company's records from when, and that his father really had sprayed and used as part of their company. Now, Roundup tended to be the pesticide that was used most often to maintain these railroad tracks. There was really minimal to no protective equipment that was used with the spraying. He would come home, and then his wife would take the clothing and do all the laundry. And this occurred really over decades. And so, we can't prove that the pesticides contributed to MSA in this patient, but it was certainly of concern, especially with the high levels that were used over decades and decades. And so that questions, well, how does pesticide exposure or chemical exposure really lead to multiple system atrophy? And one potential way is oxidative stress. So there's oxidative reactions that lead to reactive oxygen species. These reactive oxygen species are usually controlled for with natural defense mechanisms in our body and our brain. But when there's too much of these reactive oxygen species made, then that can become very destructive to neurons and any surrounding areas and almost create like a snowball type effect. We know from animal models that pesticides in particular can cause a reactive oxygen species and neurotoxins in and of themselves. So this is work in specifically multiple system atrophy. So a transgenic mice model. So this is a mouse model that's already a bit more susceptible to getting multiple system atrophy, was then treated with the mitochondrial toxin 3NP, and they have increased motor deficits and neurodegeneration compared to mice who are just treated with kind of saline solution as well. So it's suggesting that there may be a susceptibility component, that when certain individuals are exposed to these different toxins, it leads to greater neurodegeneration and the alpha-synucleinopathy pathology. So I talked about that predisposition, if you will. And so we do know that MSA is largely a sporadic disease. So for the most case, there are not clusters, and it doesn't run in families. But I want to spend a little bit of time talking next on some of the recent genetic advances in multiple system atrophy that could potentially explain why there is a susceptibility for some people for these environmental toxins. So this is work from Dr. Shoji's lab. And so he has two families. This was published in New England Journal of Medicine that had familial multiple system atrophy. And these are Japanese patients. And so you can see in the black shows the patients that are affected, MSAP, MSAP-NC, and then MSAC, but also a few family members with Parkinson's disease as well. So using these different families that have MSA, multiple family members affected, they identified mutations in a CoQ2 gene, so involved in mitochondrial function. And I think this was a really great advance. We've looked at it, or other groups have looked at it in our North American population and the European populations, and we do not find these mutations specifically linked to multiple system atrophy. But this does suggest that in this Japanese population, mutations in CoQ2 may put individuals at increased risk for MSA. With that background too, there are, constantly progressing this field. So for instance, Professor Shoji has a paper under review looking at a susceptibility loci in PLA2G4C that was identified in a Japanese MSA cohort, but the replication studies are being done in cohorts from around the world. And this particular gene is thought to affect a specific enzyme activity involved in membrane phospholipids and then alpha-synuclein biology. So there's a lot of interest in studying what particular genes may be affected or could lead individuals to be susceptible. This data on the gene-environment interactions are still pretty limited. So there's a recent case control study that found differential risk between individuals who had some of the same genetic mutations and had varied exposures to different environmental factors. So we're talking about smoking, alcohol, well water, pesticide exposure. And these gene environment interactions will be clinically relevant since the different genotypes may accentuate or perhaps improve the impact of these different environmental factors on MSA risk. We're also learning more about epigenetics and how that could be implicated in MSA through regulation of gene expression. And recent genome-wide studies have showed perhaps altered DNA methylation profiles in patients with MSA and healthy controls. And some of that may even be modified by different environmental risk factors. But there's certainly more that needs to be done to help delineate these really complicated relationships. So you can see here that these different genetic relationships, and a lot of this is taken from the Parkinson's disease litter, so more the alpha-synucleinopathies with Lewy body diseases, and linking that to different environmental factors that could contribute to the pathological findings and the neurodegeneration in multiple system atrophy. I want to switch gears a bit now and also talk about concussions. So this is another risk factor that we know plays a role in these other synuclein disorders, including Parkinson's disease. So this is a retrospective cohort of traumatic brain injury patients who were diagnosed using a VA health administrator databases. And so patients who were diagnosed with a traumatic brain injury were age matched to those without a traumatic brain injury. And this is a massive study. So 325,000 patients total, half of whom had TBI, half of whom did not. And in all of the models, any type of traumatic brain injury, from a mild one to a more moderate to severe traumatic brain injury, increased the risk of Parkinson's disease. And I think that's interesting too, that even the mild traumatic brain injury patients had an increased risk of Parkinson's disease by up to 56%. But you can see here, this risk is still low, a cumulative incidence of 0.02. And so it really took this large study population to find this association. But I think that's important to know and something that we can keep in mind as those concerned about MSA and think about concussion being a potential risk factor for the development of MSA, though it hasn't been fully studied in this MSA disease population. And you might say, well, why hasn't this been studied in MSA? And part of it is that these studies to look at environmental factors and that specifically doing a cause and effect are very difficult, in part because it involves really large numbers. So we saw 325,000 patients in that Parkinson's disease study, which could be done in Parkinson's because it is so much more common than MSA. So this direct line, so doing a cause and effect relationship really is messy in our complex world and inherently challenging. So the epidemiologic studies that are really scientists' main tool for investigating environmental risks are fairly good at identifying associations between something and the environment, yet, like I said, require these really high numbers, which makes it so difficult in MSA. And even if you have do an epidemiologic study, the statistical relationships cannot by themselves prove that one thing causes another. So that is still something that is really difficult in doing this type of epidemiologic study. We talked about the rarity MSA, and also in looking at these different studies, the research is often inconsistent, meaning that the questions asked or how you survey pesticide exposure, how you quantify pesticide exposure or different occupational exposures is difficult to do well, and especially difficult to do at the scale that we need. Also, study populations may differ. So we see that there are, subtle differences in MSAC, MSAP, and also differences in likely different environmental exposure. So someone in the Midwest may have a different pesticide exposure than, say, someone in the South, than, say, someone who lives in Japan. So that makes all of these studies hard to do if we need big numbers to really try and find different associations. So this is just my effect graphic to say that it's really hard to do a cause and effect analysis because there are so many different things that are at play in any one individual. So then trying to extrapolate to a big study population makes that even more difficult. That doesn't mean we shouldn't try and keep looking, but I think that is just something I wanted to share with the group. So I'm going to conclude now. We've talked about how there's different vulnerability of neuronal groups and how these different networks likely underlie the course and the progression in these alpha-synucleinopathies, and how multiple system atrophy is inherently different from the other synucleinopathies like Parkinson's disease and dementia with Lewy bodies. We think that environmental triggers may play a role in an individual with a genetic risk factor, yet a lot of these studies are difficult to do because of what we just talked about. And I'll also highlight then too, we still don't have known genetic causes in our North American population. I'm certainly excited with all the work that is being done in these worldwide cohorts and sharing samples to really hope that in the long run we will have better idea of which genes and which environmental toxins, if they are, may contribute to multiple system atrophy. So thank you. It's been an honor to get a chance to talk with you today, and thank you for listening to my presentation.